Okay, so for this audio lecture, it's kind of the cliff notes of antimicrobials, which is covered in chapter 28. So I've removed a lot of the individual antimicrobials, and just kind of stuck to like the kind of big picture pieces. So just be mindful of that. We did do the class activity, and there was that PDF that had a number of different antimicrobials in there. And as I mentioned in class, I would expect you to know examples of like antimicrobial that's like bacterial static versus bactericidal. I'd expect you to know examples for each of the different mechanisms of action, but I don't expect you to know every single one because this is not a pharmacology class, and you're not all going into the pathway of being doctors and such. So, all right. So, with antimicrobials, they're also called chemotherapies. When we tend to think of chemotherapies, we think of like drugs that kill cancer cells, but chemotherapies are drugs that kill cells. So for antimicrobials, that's gives us a little more information that, okay, the target, the cell that we're killing, is actually going to be microbes. And then we can even go more specific with like antibiotics, antivirals, antifungals, antiprotozoans, antihemolyphs. So again, keep in mind, chemotherapies is this very general term. It is a chemical agent that kills cells. Antimicrobials is a chemical agent that would kill microbes. An antibiotic is a chemical agent that would target bacteria. And then antivirals target viruses, antifungal target funguses, antiprotozoans target protozoans, and then antihemolyphs would target parasitic worms. Okay, so we also want to keep in mind the different properties that microbes have are going to affect what antimicrobials work against them. So in some cases, they outright kill, and we would refer to those as cidal. And in other cases, they inhibit their growth, and in those cases, we would call those static. So in both cases, whether we directly kill the microbe or whether we inhibit their growth so they can't increase in numbers, we achieve what's called microbial death, and we'll talk more about that. So there's a number of different... Now, when we look at the general characteristics of antimicrobial drugs, we can look at selective toxicity, so the ability of the drug to kill or inhibit pathogens while damaging the host as little as possible. So obviously our goal is to have in the discovery of the next antimicrobial, we want to select something that isn't going to be toxic to the individual or as little as possible. So what you will notice with the mechanisms of action, there's a lot of this us versus them. So what do they have that we do not have? And that's where we target. We also want to make sure that there's a therapeutic dose, and that's the dose that's actually required to treat the infection. Drugs are also going to have a toxic dose, and this is where the level would become too toxic for that patient. So then we would see an increase in side effects and severity of side effects. Okay. And then the therapeutic index is that ratio of toxic dose to therapeutic dose. And so all those are great, like pharmacology type terms. So when we look at side effects, those are undesirable effects of the drugs to the host cells. So just with any drug, if you look in the packet that comes with the bottle of medication, there's going to be certain side effects. Certain side effects are are acceptable. Other side effects obviously are ones that we would not want. We also want to be mindful some side effects can vary. So depending upon how individuals metabolize the drug, we could see side effects. And then when we're talking about antimicrobials, sometimes the side effects we get are because we're killing our own beneficial microbes or symbiotic microbes. And so sometimes those side effects are because we are actually affecting like the ecosystem in our body. When we look at antimicrobials, especially antibiotics, they fall into two categories, narrow spectrum and broad spectrum. So narrow spectrum is only going to target a few pathogens that have very specific characteristics, where broad spectrum is going to attack many different types of bacteria. Okay, so narrow spectrum, an example would be like penicillin. So penicillin is a cell wall inhibitor. And so if you have a thick cell wall, like in the gram positives, penicillin will target you. If you have a thin cell wall, it's not going to work against you. And when we look at like affecting just gram positive, that would be narrow spectrum. Obviously, there's a number of bacteria that are gram positive, so that may not seem narrow spectrum, but it is. And if we can look at other mechanisms of actions, like certain metabolites that target metabolites, again, that's where you might see something that's narrow spectrum. Now, with broad spectrum, it's going to target not just gram positives, but also gram negatives. So this is where the mechanism of action might be something like protein synthesis, because it targets the ribosome, and it targets a part of the ribosome that both the gram positives and gram negatives have. And so it would work on both of those. Tetracycline or doxycycline, that's a great example of a broad spectrum. Okay. And then we have cidal agents. So it could be bactericidal, microcidal, fungicidal, and that's going to kill the target. So a cell wall inhibitor, because there becomes a weak point in the cell wall because the bacteria can't repair itself, those end up lysing and dying. So that would be an example of something that's cidal. There's a direct killing because of the mechanism of action, where a static agent can be reversible. So if you decide to stop taking the antibiotic early, you haven't killed the pathogen, it just inhibits the growth. And so protein synthesis inhibitors are a great example of that, because you block protein synthesis. The microbe still has all the proteins it already synthesized, it just can't make new ones. Well, in order to grow and divide, to be in that exponential growth phase, you need to be undergoing protein synthesis. So you inhibit protein synthesis, it can't divide, but it's not being directly killed. Now, if you take something that is a static agent like tetracycline for a long time, it will start killing, but it's not a direct killing. It's just this lack of ability to make new proteins. Proteins have half-lives, and so eventually they'll end up dying. So bactericidal agents tend to have a shorter dose time, so 5 to 7 days, where bacteriostatic drugs, they tend to have a longer, so anywhere from like 10 to 14 days. There's different ways that we can assess what is an effective drug. So there's various reasons we'd want to do this. One of them, if we're just trying to assess what, in research and development, what the antimicrobial might target, we would want to know what is an effective dose that would be needed to kill the microbe or prevent growth of the microbe. And then we'd also have to do toxicity assays to make sure that for the eukaryotic cells, the human cells, it wouldn't be toxic to. It also might be that you have individuals who can't take certain antibiotics, so the standard go-to antibiotic they can't use because they're allergic. So we'd want to assess what antimicrobial we could use against that pathogen. Also, if we know there's resistance, so if we've tried a number of different antibiotics and they're not working, that's where we might do additional testing like this. So one of the ways, one of the tests that can be done is MIC, so minimal inhibitory concentration. So this lets us know what is the lowest concentration of the drug that prevents the growth. So you'd have a series of test tubes set up. They would be inoculated, and they would have different concentrations of the antimicrobial in it. So usually you do like a serial dilution, and you would then do an assessment on which tube, what concentration doesn't have growth in it. Okay. There's also the minimal lethal concentration. So that's the lowest concentration that kills the pathogen. Okay. So again, one we're trying to prevent the growth, and the other one we're trying to kill the pathogen. And then the more standard test for looking at susceptibility, again, either due to resistance or due to individuals' allergies, would be the Kirby-Bauer. So in this, Kirby-Bauer is also referred to as the disc diffusion assay. So in this, you'd put a lawn of bacteria. So you'd put bacteria on the plate, and then use this device called a hockey stick to then smear that bacteria over the whole entire plate. So when I say a lawn, it's just like the idea that like a beautiful lawn has grass growing in all locations. So you're depositing the bacteria over the whole entire surface, so you get this nice even distribution of the bacteria on it. And then using a device or forceps, you then deposit these discs. So you can see in this image the different discs that are present there. So each of these discs would have an antimicrobial, and the antimicrobial at a certain concentration. And so this allows you on one plate to test a number of different antimicrobials against that bacteria that you plated. So that bacteria could come from the patient or in a lab setting. Again, if you're trying to do assessments, because there's this bacteria that causes issues and you've got a new antibiotic, but you want to compare it to the available antibiotics, you would do the disc diffusion test. So you can see on like C, there's a nice area where the bacteria has been cleared or killed, right? So that's the reality, is if an area of the plate is clear and you can kind of see through it and there's no bacteria there, that means the antimicrobial has killed that bacteria. So that would be a good antimicrobial to give your patient. And what's created is what's called the zone of inhibition. So the diameter, so from one side of that circle to the other side of the circle, is referred to as the zone of inhibition. The larger the zone of inhibition, the more susceptible it is. The smaller the zone of inhibition, that indicates either that's not the correct antibiotic for that because of the mechanism of action, or there's resistance. So you can see in E, there's bacteria growing right up to E. And then also A, you can see how there's this it's kind of a little less clear, a little cloudy there close to the disc. So there would be some bacteria that are actually kind of resistant. And this might be something that has intermediate resistance. E would be a full resistance, or E might be the wrong mechanism of action. So if this was like a gram-negative bacteria that's growing on this plate and E is penicillin, it wouldn't be surprising that we have this very small or no zone of inhibition. Okay, so again, we always, when we're prescribing antibiotics, we want to give individuals something that is going to work. And so again, sometimes we default to the go-to. We run the culture, we get results, and then we might use the Kirby-Bauer to then switch to a different antibiotic, especially, again, if the patient's not feeling better, which would indicate resistance. All right, so the antibiotics have different modes of action. So these modes of action or mechanisms of action are really more specific to antibiotics than antimicrobials in general. So with antibiotics, again, we want to keep in mind what's different between them and us. And so when we're talking about the different structures that we see in eukaryotes versus prokaryotes, there was a couple that we talked about. So one of those is the cell wall. So we don't have cell walls, but bacteria do, or most bacteria do. And then when we look at our ribosome and their ribosome, those are different, right? So you can notice the top two are inhibitors of cell wall synthesis. So a bacteria needs to be able to synthesize cell wall in order to divide. They also need to be able to repair their cell wall. So inhibitors of cell wall synthesis block both the growth, because they can't make cell walls for two daughter cells, but also the repair process that puts weak points on the cell wall. So the membrane is going to push up against, and eventually the cell's going to lyse. And so that's why it would be cidal. Protein synthesis inhibitors target the 70S ribosome. So it can bind to either the small subunit, the large subunit, it can block the message RNA, it can block the transfer RNA. So there's lots of different more specific mechanisms within protein synthesis inhibitors. There's also metabolic antagonists. So if we look at metabolic pathways, like one of the common one that's looked at is folic acid synthesis. So bacteria need to be able to produce folic acid. We get folic acid from our diet. Eat your Wheaties, eat your fortified cereals, because that's going to get you folic acid. You can also get folic acid from spinach. So we get folic acid from our diet and also from the microbes in our gut. Bacteria have to synthesize folic acid. You and bacteria utilize folic acid in order to make proteins and DNA and RNA. And so without folic acid, you can't do those things. And so the sulfa drugs are one of the major groups of drugs that target that folic acid synthesis. So that would be an example of a metabolic antagonist. And then we also have nucleic acid synthesis inhibitors. So, again, that process of growing and dividing, you need to synthesize DNA. And so if we block nucleic acid synthesis, the bacteria wouldn't be able to grow and divide. So examples of inhibitors of cell wall synthesis, we have penicillin and also cephalosporins. Sometimes these are referred to as beta-lactams, because they have a beta-lactam ring. So if you look at the figure over here, you'll notice that there's this kind of ring structure that's kind of conserved. In this top figure, you actually have the different generations of penicillin. So there's penicillin that starts, but as you go down, you also notice there's like ampicillin, amoxicillin. So the psillins, a lot of the psillins fall under the penicillin category. Cephalosporins are very similar, but they have two side chains. So you'll notice that beta-lactam ring, and then on both sides, there are side groups. That means that, chemists, when they're trying to figure out like the next antimicrobial, if they notice there's that beta-lactam ring, they can look at those side chains, and then they can even change those side chains to come up with new generations of cephalosporins and penicillins. For some reason, the cephalosporins, especially newer generations, tend to be more broad spectrum. Sometimes in medicine, we don't care why. We don't know why. All we care is it does. So there are cephalosporins that can target the gram-negatives. And again, it doesn't really make sense because they should be blocking cell wall synthesis. But hence we're there. Some people who are allergic to penicillins can be prescribed cephalosporins. So if individuals like decades ago that were allergic to penicillin tried cephalosporins and had allergic reactions, it could have been that there was contamination of penicillins, and that contamination factor has really been removed. So again, they're seeing that some people that are allergic to penicillins can take cephalosporins. That's not always the case. Some people are allergic, like across the board, to both. One of the areas of resistance is due to beta-lactamases or penicillinases. So those enzymes actually cleave that beta-lactam ring. And so that then makes the penicillin inactive. Penicillin blocks cell wall synthesis. So if you then deactivate the penicillin, the bacteria can synthesize a cell wall, so it is now resistant and the infection can continue. Okay, so hopefully this gives you some examples of cell wall inhibitors and some information on that. Next, looking at protein synthesis inhibitors, there's a number of different categories of antibiotics that fall in into this. I've mentioned tetracycline, doxycycline. So here you can see over on the side the image of tetracyclines. Aminoglycosides also fall under that category. So like gentamicin, and we have a host of other ones. Because these block protein synthesis, because they bind to some portion of the ribosome or block the message RNA from getting into the ribosome, they tend to be broad spectrum because they're not binding to like a gram-positive ribosome. They're binding to a conserved area of the ribosomal proteins or our ribosomal RNA. And they are static because, again, as mentioned, the cells will already have proteins made. So until those proteins break down, they would be able to survive. And then looking at metabolic antagonists, so as I mentioned about the sulfa drugs disrupt folic acid synthesis pathways. Those are broad spectrum because both gram-negative, gram-positive, and other bacteria utilize the pathway in order to synthesize folic acid. So again, blocking some kind of metabolic pathway would make that the category of metabolic antagonist. Just to provide you a little bit more information about sulfas, they're very similar to PABA, and PABA is a substrate that is utilized for folic acid synthesis, and so it competes for the binding site in that the binding site of PABA, and so if sulfa drug is bound to that site, PABA can't bind, you can't make folic acid. So it's a competitive inhibition, which means sulfa drugs have to be given at a higher concentration. If you give something at a higher concentration, you are more likely to have side effects. And we do want to keep in mind that side effects are not an allergy. An allergy is an immune-driven response. So in some cases, both with penicillins and cephalosporins and other antibiotics, people suffer side effects and they think they're allergic. It doesn't, just because you have side effects doesn't mean it. So sulfa drugs, individuals, because it's given at a higher dose, are more likely to have side effects and sometimes identify then as being allergic, but again, it's just a side effect. And again, keep in mind, it's broad spectrum, so it's going to be inhibiting not just the microbe causing your disease, but all those other microbes. And for nucleic acid synthesis inhibitors, those are going to block different enzymes, inhibit different enzymes that are involved in DNA synthesis or RNA synthesis. So fluoroquinolones, they block DNA gyrase, and so that affects the ability for DNA synthesis. So the bacteria is not going to be able to grow and divide. An example of a fluoroquinolones is Cipro. And then looking at like blocking DNA polymerase, rifamycin would be example, like the rifamycins would be an example of that. So keep in mind the process of nucleic acid synthesis, both DNA and RNA synthesis, is highly conserved between prokaryotes, bacteria, and us, eukaryotes. So there is not going to be that selective toxicity where it just affects the pathogen. It will have effects to us. And so we want to keep that in mind. So even though this is a broad spectrum bactericidal, these are not your first class, your first go-to antibiotics. So I mentioned about Cipro. Cipro can have very toxic effects, lifelong effects. So it's not meant to be in every, like, a go-to antibiotic. So Cipro is known to affect the tendons. So if you have a connective tissue disorder, it's really not recommended to take Cipro. It can cause aortic dissections because it affects the connective tissue in the blood vessels. It can also cause tendons to rupture. So your calf muscle is a large muscle, and so that can actually rupture and tear. So your Achilles tendons can separate. So again, these should not be first class. They now have a black box label on them. All right, switching out of bacteria, looking at targeting antivirals. There's a number of different antivirals out there. Some of the antivirals are designed to target specific viruses, and some of these are even monoclonal antibodies that we're seeing that are being utilized. So they are meant to either block the entry of the virus, block viral synthesis, so synthesis of either viral proteins or their genome, or they block the entrance. So when we look at the mechanisms of actions for antivirals, they fall in those three categories, because those really are the three steps of viral replication. Okay. Again, if you're blocking entry, sometimes what they do is they bind to glycoproteins that are on the surface of the bacteria or of the virus, or they bind to the receptor that they target. So an example of blocking entry would be Tamiflu. So it is a neuraminidase inhibitor. So neuraminidase is one of the glycoproteins that are found on influenza. And acyclovir, that one actually is going to block the viral DNA polymerase, so that the virus can't replicate its genome. So that would be an example of blocking synthesis. So in many times antivirals, they might limit the duration of the disease or the severity of the disease. And again, that is, and they usually have to be taken early. So we see that with like the COVID antivirals. You have to take that in the first like 3 days of symptoms. After that, there's been so much viral replication that the antiviral drugs are not going to be very helpful, because really what you're trying to do is prevent that virus from being either synthesized, made, or being released, so that it can infect all these other cells. If you wait too long, there's been so many viruses replicated again, it's not going to have much of an effect. For antifungal drugs, there's less of these because funguses are eukaryotic and we are eukaryotic, so we actually share a lot of the same features. And so the cell wall would be one of the targets. There are antifungals that kind of incorporate into the cell wall and cause these weak points. But many of them, in order for them to have a therapeutic dose, it has to be such a high concentration, and that ends up being toxic to us. So now for the most part, even though fungi are similar to us, our immune systems actually does a very good job of keeping fungal infections in check. Usually, if you have uncontrolled fungal growth, that might be an indication that something else is going on with your immune system. And then we have antiprotozoan drugs that target protozoas. So a lot of the mechanisms of actions aren't actually known. It's one of those like, hey, they kind of work, so people get better if they take them. So we'll worry about that later on. So maybe someone will find some research money and research that. We do know that some antibiotics seem to inhibit protein synthesis in protozoans. So there's some use of antimicrobial antibiotics for protozoan infections. There are antibiotics that also cause kind of activate the immune system, so they can affect inflammatory pathways. So whether that's the mechanism that's also at play, whether it's somehow kind of priming your immune system and getting, upsetting your normal microbiota, so they actually are producing antimicrobial components, that would be an amazing project to look at, right? But one of the antiprotozoan drugs that's used is chloroquine. So chloroquine is used for malaria infections, but there are a number of other ones. All right, so lastly, I want to talk a little bit about resistance. So there are a number of different ways in which drug resistance can occur, okay? So they can be intrinsic. So they occur due to the properties of the microbe itself. So Mycoplasma is resistant to beta-lactam antibiotics and other cell wall inhibitors because they don't have a cell wall, right? So an intrinsic is it's really the wrong antibiotic. The mechanism of action is not going to work because they don't have that feature. They don't have that characteristic that's being targeted. Acquired is where there's a change in the genome. So acquired, when we look at resistance of like methicillin and Staphylococcus aureus, that would be an acquired one. So they've picked up a plasmid, they've picked up a gene, or there's been some kind of mutation to their genome. So they now express an enzyme that actually is able to deactivate the antimicrobial. Okay, so again, acquired, there's been a change. It's not that we picked the wrong antibiotic. So there are drug tolerant bacteria. So those are known as persisters. So they lack the mechanism for antibiotic resistance and ignore the presence of antibiotics. And so what can happen, and usually that's because they're embedded like into a biofilm. And so biofilms have a matrix to them. And so that can affect whether you can actually get that antimicrobial pass into that actual organism. Also, some bacteria, as we mentioned, can actually hide into tissues and into certain cells. And so again, they're not resistant, but again, the antibiotic can't get to them. So looking at the mechanism of drug resistance or antibiotic resistance here, we can see here we've got our bacteria. There's a number of different mechanisms that can be at play. Now this figure has 4 different mechanisms, but really there's only 3. Okay, so the first one that's at the bottom of this bacteria is that there's an altered antibiotic target. So again, if we think about what the drug has to bind to, there could be some kind of mutation that's arise that leads to that binding site not being available. So here we've got our ribosome, and maybe one of the ribosomal proteins have been mutated, and now the drug can't bind to the ribosome to inhibit protein synthesis. Now for 2 and 3, you have enzymes that are either going to degrade the antibiotic or alter the antibiotic. So in both cases, we end up having these enzymes, and these enzymes are going to affect the ability for the antibiotic to do the thing it's supposed to do. So if we look at beta-lactamases, in some way they kind of work the same. So a lot of times this category gets grouped together, and it's just an antibiotic modifying the antibiotic with an enzyme. So beta-lactamases are going to cleave that beta-lactam ring, and that deactivates the antibiotic. Okay, and then the other way is that the permeability of the antibiotic is affected. So a lot of times what will happen is the bacteria will acquire a gene for a pump, and that pump then is going to actually pump the antibiotic out. Now, when we're thinking about something that's blocking protein synthesis or nucleic acid synthesis or a metabolic pathway, it has to get inside the cell, right, to have that function. So a lot of these antibiotics have to get across the membrane and then into the cell. If you are a bacteria and you just end up pumping that back out, it never has an opportunity to bind to the target, so it can't have an effect. Okay, so you'll notice that when we're looking at enzymes that are altering the antibiotic, and when we're looking at the pumps, those are acquired through plasmids. So plasmids are going to have genes, these kind of luxury genes, that give that bacteria that resistance. When we're looking at the altering of the antibiotic target, that's because there's been a mutation that has arisen that leads to a change of the binding site of the target. So there's some ways to overcome drug resistance. So we want to make sure that we're giving drugs at an appropriate concentration to destroy the susceptible microbes. And so keep in mind that we are a mosaic of different types of microbes. And then even within the species, you can have, if a spontaneous mutation arises, that microbe could be now more or less susceptible to the antibiotic. So again, we want to make sure that we're killing the microbes, so we don't allow for any of those intermediate or resistant microbes to persist. Okay, that also means the course of time that the drug is given, right? So we want to make sure that the complete dosage is given, so as prescribed. So sometimes what happens, patients start to feel better. They say, oh, I can stop taking my antibiotic and I can save these for the next time I get sick, right? So everybody's got that family member who's like, oh, I got some medications. Yay, take this, right? So they really should have taken the full course. They should have taken what they was actually prescribed. And we also want to make sure we're not taking other people's antibiotics, because if we're taking drugs when it's the incorrect drug, we're just leading to resistance. So using drugs only when necessary, and then using the correct ones, right? We also want to, in some cases, give more than one drug at the same time. So there are drugs that are actually a combination of either two antimicrobials or an antimicrobial and something that deactivates the enzyme. So augmentin is an amoxicillin plus an acid that deactivates the beta lactamases that might be present, so that can override the resistance that the bacteria might have. Okay. And then we also want to keep thinking about what are the new drugs, right? So what is the potential new drugs? One of the areas that's being looked at for antimicrobial therapies are quorum sensing and drugs that block quorum sensing. So Bonnie Basler is one of the leading researchers who is researching quorum sensing between bacteria, and again looking at, can we develop blockers of quorum sensing, because a lot of the pathogenic behaviors of bacteria are due to quorum sensing, and we'll end up talking about that in our next unit. Also looking at using bacteriophages to treat bacterial diseases. So bacteriophages are viruses that infect bacteria, and so if we develop bacteriophage that have kind of these kill signals in them or kill genes in them, that can actually deliver that to bacteria. We want to make sure that we're picking bacteriophages that are specific to the bacteria that's causing the disease, so that we don't end up, affecting our normal microbes. Okay. So hopefully that provided you some of the background information about antimicrobials, some of the terminology, and even though I didn't have tons of examples, hopefully you have examples of each of the different mechanisms of actions, and enough that you can feel comfortable answering some of those questions on the exam and such. If you do have any questions, don't hesitate to contact me, and have a great day.